So if you remember from a previous lectures, we had discussed DNA cloning, and I told you that DNA cloning is a technique for amplifying and or expressing DNA fragments. And there are two basic ways of cloning a DNA. First, of course, is the in vivo or cell-based method of DNA cloning, where you take your DNA of interest, ligate it to a vector, the recombinant vector is transformed into a host cell, and then, of course, within the host cell, the vector is going to autonomously replicate, increasing the number of copies of your DNA of interest. And also, as the cell divides, the number of copies of your DNA of interest multiply again. So this is also a kind of exponential amplification that happens here. The advantage here is that apart from amplification, you can also express your DNA of interest in vivo or cell-based method. The other method is what is known as the in vitro or PCR-based method. In this method, the DNA can only be amplified. It cannot be expressed here. So that's the basic difference between the two. You can have an expression of your DNA in vivo or cell-based method, but in case of PCR or polymerase chain reaction, which is an in vitro method, where you're doing it outside a living cell, in this case, you can only amplify your piece of DNA. You cannot really express it, right? So the polymerase chain reaction combines the two basic principles of biotechnology. One, that is nucleic acid hybridization, and second, replication. The two are combined together to give you an amplified copy of DNA. And it is a very basic and versatile technique and has been modified several times to give rise to other types of applications of polymerase chain reaction, which we'll discuss as we move into the lecture. So as an activity, I want you to name any two techniques based on the principle of nucleic acid hybridization. So here you are, you have a piece of DNA here. If you can see, this is a double-stranded DNA, and the two strands are stabilized by the hydrogen bonds between adenine and thymine and cytosine and guanine. And you can see that the two strands are anti-parallel. The 3' end is represented by the arrowhead here, and the 5' end is represented by the blunt end. So you can see the 3' end of the top strand is here, and on the opposite strand, it is the 5' end that is opposite to the 3' end of the top strand here. So this is the two strands are running anti-parallel to each other. They are bound into a DNA double helix, which is the basic model of DNA structure. Now, if you provide heat to this DNA molecule, the two strands are going to denature. So here you are, you provide some heat to it around 95 to 96 degrees centigrade. The hydrogen bonds between the two strands will melt down, and that will result in separation of the two fragments. Now, as you allow the temperature to come down, the two strands will be able to find each other and then reassemble as a DNA double helix stably. So that is the process of nucleic acid hybridization, where two complementary strands which are running anti-parallel to each other will be able to find each other in a mixture of other DNA strands and stably hybridize with each other and form a DNA double helix. So this is what happens here when you bring up the temperatures. The two strands again come close to each other, form hydrogen bonds and stabilize, right? So this is your nucleic acid hybridization. The nucleic acid hybridization is a phenomenon in which single-stranded nucleic acid molecule can find an anneal to its complementary nucleic acid molecule in a mixture of other DNA strands as well. So this could be happening in isolation, or this could also happen when there are other DNA strands as well. And here I show you two strands of equal length. It can also happen between a strand that is very long and oligonucleotide that is very short, as happens in the case of hybridization of primers in case of PCR. So here you are, this is your long DNA strand. Let's say this is genomic DNA, and you have an oligo that is just about 18 to 27 bases. The oligo will be able to scan through the entire length of the longer DNA strand and find region of its complementarity and then form hydrogen bonds and stably attach to this position here, right? So this is the idea behind nucleic acid hybridization. Nucleic acid hybridization will happen between two complementary strands. The two complementary strands may not be of equal length, and still the hybridization would happen, and it will be stabilized by the hydrogen bonds between the complementary nucleotides. So this oligo, which is around 18 to 27 bases length, which is going to scan through the larger DNA strand and find its region of complementarity and hybridize to it, is called a primer. So primer is a small 18 to 27 bases single-stranded oligonucleotide, right? And in PCR, you use two primers to do the amplification. The principle of hybridization is central to many techniques in like polymerase chain reaction, where the two primers are going to first hybridize, and then by the use of DNA polymerase, the 3' ends of these primers will be extended. Southern and northern hybridization, which we'll do later, as also in microarray. Let's now look at polymerase chain reaction. The technique was developed by Caramelis, and he was awarded a Nobel Prize in chemistry in 1993 for this remarkable technique that he had developed that allowed us to amplify a piece of DNA in vitro. So the first important characteristic is that this is an in vitro method of amplifying a piece of DNA. A specific region in the template DNA marked by forward and reverse primers is amplified. The amplification here is exponential, which means that every single cycle of a PCR, the amount of DNA is going to become twice of the previous cycle, right? And therefore, the series that is followed here is 2 raised to power n, and we'll also examine this in details as we move along. PCR happens to be one of the most fundamental, versatile, and indispensable technique in biotechnology. And the basic utility of PCR is that it allows for amplification of the original DNA, which may be in very small amounts to begin with, or you may have a very large original DNA of which you only want a part to be taken downstream for the next experiment. So that can be done using primers, specifying the region that you want to amplify. And what has made PCR more versatile is the small tweaks that have been introduced into the conventional polymerase chain reaction method that allows you to use it in various diverse array of PCR applications, including reverse transcriptase PCR, real-time PCR, multiplex PCR, randomly amplified polymorphic DNA, allele-specific PCR, bridge PCR, emulsion PCR, etc. Of these, bridge PCR and emulsion PCR are specifically used in next generation sequencing, and of course, reverse transcriptase PCR and real-time PCR will do in details as we move along in the lecture. So let us now look at how the principle of nucleic acid hybridization is used to amplify a specific region in a given piece of DNA. Right? So now let's look at the polymerase chain reaction and the actual reaction itself, and we are into the first cycle of the polymerase chain reaction. So the basics first, you have your starting template DNA, which is a double-stranded DNA, which is in the form of double-stranded helix. So the two DNA strands are wound over each other and are stabilized by the hydrogen bonds between adenine and thymine, and guanine and cytosine on the opposite strands. So this is your directionality. The arrow indicates the 3' and the blunt end indicates the 5'. You have two primers, a forward primer and a reverse primer. So the forward primer, if you look here, is basically having the same sequence as the red strand here or the top strand, and will actually hybridize to the bottom strand, which is complementary to the red strand. Likewise, the reverse primer here has exactly the same sequence as the blue strand or the bottom strand, and is going to actually hybridize onto the red strand here, which is complementary to the sequence carried by the reverse primer. So the region of the DNA that is going to be amplified is marked by the 5' end of forward primer and 5' end of the reverse primer. So this is basically the region between A and B that is going to be amplified by this primer set here, right? Next, the forward primer has the same sequence as the plus or the red strand, and therefore anneals to its complementary strand, which is the blue or mantle strand. Likewise, in the reverse primer, the sequence is the same as the blue strand, and this is going to bind to the complementary strand, which is the red strand here, right? So the sequence is same as the blue or mantle strand, and it anneals to the plus or the red strand here. So in the first step of the PCR reaction, the DNA is denatured. You increase the temperature to around 94 to 96 degrees centigrade, during which time the hydrogen bonds, being weak bonds, are melted, and therefore the two strands separate out. Once the two strands are separated out, the temperature is brought down again to somewhere around 55 to 65. At this temperature, the primers are going to anneal to the template strands. So here you are at this stage. So at the annealing stage, the primers are placed like this in the PCR. So this is how the primers get placed at the annealing stage. So this is after denaturation and then bringing back the temperature to 55 to 65 degrees centigrades. You have your forward primer, which is now hybridized to the blue strand at the position where it is absolutely complementary. Likewise, you have the reverse primer, which is now hybridized to the red strand at position where it is absolutely complementary to the red strand. And the primers, as I've shown you, they scan through the entire sequence and find regions of complementarity and then become stably hybridized in those complementary regions. Now, of course, the next important innovation in PCR is that you have DNA polymerase in the solution. If you see now, for a DNA polymerase to extend a strand, you require a template. You already have a template here for the reverse primer. You already have a template here for the forward primer. And then now the DNA polymerase can extend the 3' end of the reverse primer and the forward primer to add complementary nucleotides based on what is present on the template. So this is how it happens. For the forward primer and the reverse primer, the reaction happens simultaneously, but I'm showing you in steps here. So the forward primer gets extended based on what is present on the forward primer gets extended by addition of complementary nucleotides based on what is present on the template strand, that is the blue strand here. So here is your DNA polymerase, which is going to extend your complementary nucleotides at the 3' end of the forward primer. Likewise, simultaneously, the reverse primer also gets extended, and you have addition of complementary nucleotides here. And this goes on beyond A and B, if you notice, because there is nothing here to stop the reaction at point A or at point B. Only thing that is going to stop this from getting extended is the processivity of the polymerase or the extension time that is given. So you give a specific extension time during which time the DNA polymerase is going to extend your 3' end of the forward and reverse primers based on the nucleotides that are present on the template and add complementary nucleotides to the 3' ends of the forward and reverse primers. So here you are. This is your first cycle of amplification. Let me just take you through it one more time. So you had a template DNA strand here, and you had a primer set. The primer set forward primer has exactly the same sequence as in the red strand, and the blue primer has exactly the same. Reverse primer has exactly the same sequence as is present on the blue strand. So when you denature your DNA and allow for annealing, the two primers then attach to their complementary strands or hybridize to their complementary strands. So the reverse primer hybridizes from the red strand here, like the forward primer hybridizes onto the blue strand here. And then, of course, in the presence of DNA polymerase, the 3' end of the reverse primer and the forward primers are extended simultaneously. And this results in your first round of amplification. So you started with 1 molecule of DNA. Now you have 2 molecules of double-stranded DNA after the first cycle. Also, the strands are well beyond the actual designated boundary of A and B, which is where the 5' ends of the primer of the forward and reverse primers are actually present. Right. So this is the start of the second cycle. You already have 2 double-stranded DNA molecules now, and now you denature again. So when you denature, now you have 4 strands now. So if you see here, these are the 4 strands after denaturing. You again bring down the temperature to 55 to 65 degrees centigrade, where the primers can hybridize to their complementary sequences on these templates here. So here is your primer annealing. So this is how it happens. So the reverse primer finds its complement on the red strand here. Likewise, the forward primer, which has a sequence exactly similar to the red strand, finds its complement on the blue strand and anneals here. And now again, you have the extension by DNA polymerase, which happens at around 72 degrees centigrade. We'll go into the temperature cycle once you've done this basic concepts of how the polymerase chain reaction works out. So here you are. Now you get your extension again. So reverse primer gets extended, likewise forward primers get extended. All these amplifications are happening simultaneously. The extensions are happening simultaneously. So after the end of second cycle, you have now 4 double-stranded DNA. You started with 1 in the first cycle, you have 2 in the second cycle, you have 4. Likewise, in the third cycle, you'll have 8, then 16, then 32, and so on and so forth. And we'll also discuss this progression as we move along. One thing that is clearly noticeable here is that after the second cycle, you have at least two strands that are exactly within the boundary of A and B. So if you see here, these are your two strands which are exactly the size of the amplifon that you expect when you use A and B primers. This is happening because at this point, the forward primer gets extended. There is no template beyond point B now. So therefore, no further extension happens, even if the extension time is longer. Likewise, for this reverse primer, when it starts extending, there is no template available beyond point A. So therefore, it stops extending here. And so after the completion of the second cycle, you start getting DNA strands of exactly the size of the amplifon that you expect. So now as an activity, let me ask you a simple question here. So if you start a PCR with one molecule of DNA, then how many DNA molecules will be produced after 5 cycles? And in case you're not able to get the answer straight away, then you can basically take down a pen and pencil and start doing it cycle-wise. This will also give you the conceptual clarity and also the correct answer at the end, right? And 5 cycles is not too many to draw. Okay. Or else I've already given you the answer in the lecture, and of course we'll be doing it again in the later slides. So now let's look at what is the correlation between the cycle number and the amount of DNA that you finally have. So if you start with the 0th cycle where you're starting your PCR, you have one DNA molecule to start with. This can be represented as the 2^0, or you're starting with one DNA molecule at the 0th cycle where your PCR has not yet started. Then when you complete the first cycle, you have two DNA molecules now. So this is 2^1. After completion of cycle 2, you have 4 DNA molecules, which is 2^2. After completion of third cycle, you have 8 DNA molecules, which is actually 2^3. And after completion of fourth cycle, you have 16, which is 2^4. 5 cycles, 32, 2^5. And if you extrapolate this to n number of cycles, then it is finally the amount of DNA molecules that you'll have in your solution would be 2^n, right? So this is the simple exponential formula that we follow. So after n cycles of PCR, if you're starting with one molecule of DNA, you will have 2^n molecules of DNA, right? Now, what I want you to think upon is, let's say you start with X molecules of DNA. So if you start with X molecules of DNA and run the PCR for n cycles, how many DNA molecules will be formed on completion of PCR? That's the question that I want you to think upon. And I'm sure you will be able to answer this. And of course, we'll discuss the answer in the next few slides. So let's talk of the basic exponential amplification rules. Rule number 1 is that the total number of DNA molecules doubles every cycle of PCR, which we've already discussed in the previous slide. If you start with one DNA molecule, then after n cycles, you will have 2^n DNA molecules. And the third important rule, which I had asked you in the previous question also, is if you start with X DNA molecules, then after n cycles, you will have X into 2^n DNA molecules. So these are the 3 basic rules of amplification that you must remember. 1, it doubles up every cycle. Second, if you start with one molecule of DNA, you have 2^n molecules after the nth cycle, after the completion of nth cycle. And if you start with X molecules of DNA, then it becomes X into 2^n. So now again, as an activity, I ask you a simple question. If X number of DNA molecules are formed in n cycles, in how many cycles would the number of DNA molecules become 2x? Take a pause, think, and then answer, right? This is simple, but this is tricky again. So here is your 30 seconds starting now. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. So the answer to this would be the very next cycle. So this would be n+1 cycles. Remember, the DNA is going to double up every cycle. So if it is X number of amplifications at cycle n, it will be 2x at n+1 cycles, right? So let's now look at the PCR steps, and there are three major steps in PCR. One that is known as initial denaturation, where you are going to denature your DNA. Then you have a cycle step. This is itself built up of three sub-steps, and we'll see how these steps function. And then you have a final extension step. And after the completion of PCR, usually the samples are left at 4 degrees centigrade. So you can, in case you're setting up a PCR late at night, then in the program itself, you feed that it has to be kept at 4 degrees centigrade for infinity. So in the morning, when you come back, you can take off your samples and transfer them into the refrigerator, right? So here you are, you start with initial denaturation. So this is the first step, and denaturation happens at really high temperature of around 94 to 96 degrees centigrade. Depending on what is the original template that you're using, the time of denaturation would be around 5 to 10 minutes. If it is human genomic DNA, the time would be around 10 minutes because it is a large DNA, 3.2 billion base pairs. So the time required for denaturation would be a lot more. If it is a plasmid for that matter, then of course you know that it is not going to be more than 10 kb, even if it is a recombinant plasmid. So more than around 2 minutes would be sufficient to denature that. Next, we come to the cycle steps, and cycle itself is divided into three basic steps. First, of course, is what is known as cycle denaturation. You have already denatured your original DNA template, and in cycles, you're majorly going to denature the amplicon, right? Remember, after second cycle, you will be able to get DNA strands that will be exactly the size of the amplicon, and that is what is going to be progressively amplified. So therefore, cycle denaturation is of a smaller time of around 1 minute at 94 to 96 degrees centigrade, of course. Then after you have denatured, you now want your nucleic acid hybridization to happen. The primer should be able to hybridize to its complementary strand. So therefore, the temperature is brought down. This temperature is around 55 to 65 degrees centigrade to ensure specific annealing, and the time for hybridization is around 30 seconds. And then, of course, you raise the temperature again for extension. So now your DNA polymerase comes in and extends the 3' ends of your primers. This happens at usually around 72 degrees centigrade. And the time for extension again depends on what is the amplicon size. Most commonly, when you're using a normal Taq polymerase, you would not be able to extend beyond 1.5 to 2 kb. So for that extension, 1 minute is sufficient. But in case you're doing a longer PCR, we are amplifying, let's say, 9 kb, 10 kb, or 20 kb, or 30 kb, then depending on long PCR mix that you're using and the enzymes that you're using, you would have a slightly longer extension time. And then after you have done your extensions, the last step in your PCR is what is known as final extension. Final extension again happens at 72 degrees centigrade. And usually the time given is around 10 minutes. So whatever ends that were not extended earlier now finally get extended. So this finishes your actual PCR cycle. The samples can be stored at 4 degrees centigrade, and the PCR has a refrigeration capacity where the temperature can be set to 4 degrees centigrade at infinity so that the samples are do not degrade, right? And then, of course, the number of cycles that you do, usually you do anywhere between 30 to 35 cycles of PCR, and that is enough to give you 2^30 or 2^35 molecules if you start with 1 molecule of DNA. So next we look at the reactants that are required for setting up a PCR reaction. First, of course, is your template DNA. This has to be present in a concentration of 50 to 100 nanograms per microliter. This is the DNA, a subregion of which is going to be amplified depending on the 5' ends of forward and reverse primers. Then next you require a reaction buffer. The buffer is to maintain the pH and also to provide the salts that are required for the reaction to occur. This is to provide the ionic strength and buffering capacity needed during the reaction. The composition of the buffer includes stress HCl, ammonium ions, potassium chloride. Magnesium ions may be present in the buffer or they may have to be added separately. Bovine serum albumin is an additive that is added to ensure efficacy of the polymerase chain reaction. Then, of course, if the magnesium ions are not present in your buffer, they have to be added separately because magnesium is the metal ion that is required for action of DNA polymerase. So unless magnesium chloride is added, it will not be, the DNA polymerase would not be able to function or extend the 3' end of the primers. So therefore, magnesium chloride is very important, and the concentration of magnesium chloride must be anywhere between 1.5 to 3 millimolar. In many cases, you may have to standardize the concentration of magnesium chloride. Most commonly, 1.5 millimolar concentration is good enough to give you a specific amplification. Then DNTPs, of course, have to be added because these are the ones that are going to be added to the 3' ends of the primers to extend the complementary strands. And DNTPs are present in equimolar ratio. The final concentration should be around 200 micromolar each. And after that, you also have primers, forward and reverse primers. Both have to be added in equimolar concentrations. And the final concentrations of the primers can be around 0.1 to 0.5 millimolar each. And then, of course, DNA polymerase has to be added to ensure that finally the polymerase chain reaction happens. And usually around 1 to 2 units of DNA polymerase are added per 25 microliter of reaction. The most common DNA polymerase that is added for conventional PCR is Taq polymerase, but there are other polymerases as well, and we'll discuss that in a later slide as we move along. And then, of course, the final volume has to be made up to around 20 to 25 microliters, and this is basically made up to the final volume by addition of autoclave double distilled water. And then in special cases where you have high GC content in the template, you may also be required to add additives like dimethyl sulfate, which may be added if the template has high, greater than 60% GC content. So now let's look at some of the equipment that is used for setting up the PCR reaction. So here you are. This is your PCR tube. This is a single tube, usually 50 microliters in volume. And usually you set up reaction around 20 microliters or 25 microliters of reaction is set up here. Then when you have large number of samples, let's say close to 96, in this case, the maximum number of samples that can be taken up here is 96. You have 8 rows and 12 columns, so 12 8s are 96 is what you have. You can parallelly put 96 samples in this plate, and then the plate may be finally transferred to a thermocycler or a PCR machine. The technical name for PCR machine is thermocycler because essentially what this machine does is to alternate the temperatures as per the specified time in the program. So this is the PCR machine, and here if you see there are 2 slots for taking up the samples. You could be placing individual PCR tubes here, or you could be placing an entire tray into the slot here. So there are 2 slots of 96 each. So in this PCR machine, if the program is the same, 192 samples amplification can be done in one single run of the machine, right? So let's now look at how a PCR result is analyzed. So first important point in analyzing a PCR result is after the PCR reaction is over, the samples and the positive and negative control have to be loaded onto an agarose gel. So here I show you an agarose gel which has been electrophorosed, and you can see the amplicons traveling a certain distance on the agarose gel. So what I'm interested in here is to know what is the size of my band. To ascertain the size of my band, I run it along with what is known as a molecular weight marker or ladder. So what is a molecular weight marker or ladder? Molecular weight marker or ladder is basically a pre-calibrated and known mixture of DNA fragments whose sizes are already known to us. So for example, here in this case, the lowest band is 100 base pairs. This one is 200 base pairs, 300 base pairs, 400 base pairs. 500 base pairs is in higher concentration, so comes in slightly bolder here. Then 600, 700, 800, 900, 1000 again is in high concentration, so comes in slightly more bold. And then you have 1100 and 1200. So this ladder profile or the molecular weight marker profile is specific to the company from which you buy the ladder. So therefore, you must also, when you make your gels, you should note down which company ladder has been used, because some other company may have a slightly different ladder profile than what is shown here, right? So this is important. This is your negative end, this is a positive end, and since the DNA is negatively charged, it moves towards the positive end here. So this is the direction of movement of DNA on electrophoresis. So the PCR results are analyzed by agarose gel electrophoresis, which is again the basic form of electrophoresis. It is a horizontal gel that we use instead of a vertical gel that we use in page electrophoresis. The PCR products are loaded in wells of the agarose gel. So this is your, at the top here would be wells where your PCR products are loaded. Alongside the samples, you must also have a positive control and a negative control. What is a positive control? So positive control is basically a sample where you know that you will definitely get a desired amplicon. Negative control is where you know that you would not get the desired amplicon, and most commonly this happens because you do not add the DNA template. If there is a contamination in master mix, then in that case, even a negative control, you'll get a band. When you do not have a band in negative control and you have a band in positive control, then you know that your PCR reaction has gone right. And now you can check for what is the amplicon status in the samples that you've tested. So this is basically very important for any experiment. Setting up a positive control and a negative control is just as important as setting up the reaction for the samples. And whenever you observe the results, remember, first you have to see whether you've got the positive control right and whether you've got the negative control right. So in negative control, in a PCR, you will not get a band. In a positive control, in a PCR, you would definitely get a band and of the expected size. Once these two are in agreement with the expected pattern, then you move on to check your samples. And here in this case, if you see, as for the ladder here, we get bands around 200 bases, right? So this is for all the samples that we've tested, we've got an amplification at around 200 bases. So the PCR products are loaded in wells of the agarose gel. The first or last or both wells are loaded with molecular weight markers. So usually if it is a small number of samples, you would put up a ladder at the right end. If there are too many samples, it is advisable to load ladders at both the ends. Then, of course, positive and negative controls are loaded alongside, and I've already told you positive and negative controls are the most important components of your experimental setup. Unless the patterns observed in positive and negative control are exactly as per what was expected, you cannot go and analyze your sample results. The size of the amplicon and base pairs is identified with reference to the molecular weight marker. So this I've already told you here. So this is how you analyze your PCR gel. So a PCR gel would tell you two things. One is the size of the amplification, and second, it can also tell you the relative amount of amplification that has happened across samples. For example, here, there's a slightly lower amplification as compared to the amplification you obtain in the same number of cycles in this sample here, and that is probably because of the lower quality of DNA for this sample as compared to the quality of DNA here, right? So this is again very important. Let's now talk of a direct application of PCR and what we have studied till now. So if you remember from the previous lectures, we had talked about ligation reaction, where you are ligating your insert to a plasmid vector. So you open the plasmid vector and then you add your ligase so as to make a recombinant plasmid, right? And once the ligation reaction is over, it is always good to confirm the success of your ligation reaction before moving to transformation. The most common preliminary as well as a confirmatory test that you can do is to do a PCR check. So you amplify using primers that are on the vector and see whether you get the expected band size or not. If you get the expected band size, then of course you do a confirmatory test by getting the band sequenced using the primers that you have used for amplification of this band. So let's now look at the question here. So the question is, if the PCR primers to check the ligation are 100 base pairs apart in the vector sequence and the insert size is 400 base pairs, what will be the observed band sizes for recombinant and self-ligated plasmid after PCR of the ligation mix? So here is what you have in the question. You have two plasmids after the ligation reaction, one that is a self-ligated plasmid, and you have a recombinant plasmid here. So here is how the primers are placed in the self-ligated plasmid. You have a primer, this is a forward primer, this is a reverse primer, and they're separated from each other by 100 base pairs of sequence. The primers are going to anneal at the same position on the recombinant plasmid as well. So here they are. But now, apart from the 100 base pairs that they initially had, they also have 400 base pairs of extra insert DNA in between them, right? So therefore, now the band sizes between the recombinant plasmid and the self-ligated plasmid will be different on PCR. So if you look at the expected band size here, because there is no extra insert sequence, you will have a 100 base pair band because that is the distance that separates the two primers on the self-ligated plasmid. So this would be 100 base pair band. Here, apart from the 100 base pairs that is pre-existing, you have additional 400 base pairs, So here in this case, you'll get a permanent base pair band, right? So the amplicon may be then sequenced for further confirmation. Let us now look at the properties of DNA polymerase that are required for efficient amplification. The first important property of an enzyme to be used for polymerase chain reaction is it has to be thermostable, because the temperatures fluctuate very often and very frequently in a polymerase chain reaction. And this is a reaction that is happening at relatively high temperatures of 55 or above. The denaturation happens at around 94 to 96 degrees centigrades. The annealing happens around 55 to 65 degrees centigrades. The extension happens at around 72 degrees centigrade. So these are all high temperatures. Therefore, the polymerase that is to be used for PCR must have a high thermostability. It should be able to survive high temperatures and frequent changes in temperatures as well, right? So the second important property of DNA polymerase here is what is known as fidelity, which is basically an expression of error-free replication. So the enzyme should be able to add complementary nucleotides at the 3' end of the primers faithfully and without making errors, right? So specifically when your amplicon is to be taken forward for expression. So a single nucleotide change can result in change in the codon and essentially a change in the final protein that is getting formed. So therefore, it is important that the DNA polymerase that is used must have high fidelity. The third important property that DNA polymerase must have is what is known as processivity, which refers to the number of nucleotides being processed in a single binding event. So once the DNA polymerase attaches to the 3' end of a primer, how far can the 3' end of the primer be extended is what is processivity, right? So this basically defines the maximum size of the amplicon, and usually when you use a normal Taq polymerase, you cannot extend beyond 1.5 to 2 kb. If you want to have longer amplicons or larger amplicons, then of course you have to use special polymerases or a mixture of polymerases. So here are some of the most common DNA polymerases that are used for polymerase chain reaction. First, of course, is Taq polymerase, which is the most common DNA polymerase that is used for polymerase chain reaction. It is obtained from Thermus aquaticus, and it is thermostable, and of course the processivity is around 1.5 to 2 kb. Then you have PFU, that is derived from Pyrococcus furiosus. It is a DNA polymerase with high fidelity. Then you have VENT. This is derived from Thermococcus literalis. It has moderate fidelity. Then you have special proprietary enzymes that are used for long-range PCRs, for example, platinum super 5-2 DNA polymerase. This is a high fidelity long range and can amplify fragments up to 40 kilobases in length. Next, we come to the properties of primers, which is one of the most critical components of your PCR reaction. Unless the primers have been designed well, you may not get the desired amplicon or a specific amplification. So here, if you see, you have two primer pairs. You have a forward primer and you have a reverse primer. So this is a forward primer scanning through the template to find region of complementarity and finally hybridize it at position A here. Likewise, the reverse primer would again move through and scan through its own complementary template, and then of course hybridize to a region where it is absolutely complementary. And then as you add DNA polymerase, the extensions would happen now. So this is for the forward primer, and simultaneously the reverse primer would also get extended in the other direction. And we've done this earlier. So the important part here is that the most critical component for a successful specific PCR amplification is the PCR primers. The forward and the reverse primers work as pair and in tandem to give you exponential amplification. There are certain basic rules that must be followed in primer designing to ensure that you have efficient amplification of the specific region that you wanted to amplify. Then a conventional PCR requires primers to anneal at only one unique location on template, yielding a single specific amplification. So this is again very important, that most commonly in a conventional PCR you would want a specific amplification, and for that it is important that the primer goes and anneals to one and only one position on the template DNA. And then for specificity, the primer length, composition, and primer melting temperature are of critical importance. So we will now discuss how the specificity is affected by the primer length, primer composition, and the primer melting temperature as we move into the lecture. So let's look at what do you mean by specific annealing in the first place. So here we are. This is your template DNA. This is the primer here. The arrow represents the 3' end of the primer. The blunt end represents the 5' end of the primer. And as you can see here, there is perfect complementarity between the primer and the template DNA where it has gone and hybridized. Every single nucleotide on the primer has a complementary nucleotide on the template DNA. So this is what is known as specific annealing or perfect annealing or a perfect priming. And in conventional PCR, we are expecting a specific annealing of both the primers to their respective targets and a specific amplification. So this is what is known as a specific annealing or perfect priming. In conventional PCR, we expect perfect priming to happen for both the primers, giving you a very specific amplification. However, you can also have non-specific amplification. So one example of non-specific amplification is shown here. So here in this case, as you can see, the 3' end of the primer and the 5' end of the primer are well anchored and have complementary nucleotides on the template DNA. However, in between there are nucleotides between the primer and the template DNA that are non-complementary. So this is an example of non-specific annealing, where the annealing is not perfect. This type of annealing can be used to introduce mutations into the coding sequence. Then you could have other types of non-specific annealing. For example, here in this case, as you can see, the primer is extra long, and there is perfect complementarity between the primer and the template until a certain distance, and beyond which towards the 5' end there is no complementarity. This is because this 5' end has been added extra to the primer and contains the recognition sequence for the restriction site that you want to add to your insert here, right? So this type of primer can be used to generate specific restriction sites on the insert that facilitate ligation of the insert. And then finally, you could have another type of non-specific annealing, which is this. So here in this case, the 3' end is non-complementary. The terminal nucleotide does not have a complement on the template DNA. There is complementarity towards the 5' end and substantial complementarity as well. But this primer, because the 3' end is not complementary to the template DNA, this primer will not give you any amplification, right? And such a primer design can be used to ascertain the presence of single nucleotide polymorphisms. The higher the temperature, the more specific the annealing, and therefore, if you have higher annealing temperatures, you can avert non-specific annealing. And usually when the primer pairs come to you, they have to be standardized to find out the best temperature where they can give you a specific amplification. And this is done by setting up a gradient temperature PCR where the primer pairs are tested at different temperatures within a given range to see which temperature gives you the expected amplicon without any non-specific annealing. So the first important property for getting a specific amplification is that the primers must be unique. The primer must have only one perfect complementary site on the template DNA. Right? So as far as possible, the perfect primer complementary sequence must appear only once in the template DNA. And to ensure that this happens, you have to have a primer which is not less than a specific length. And in this case, empirically, it has been found that it is equivalent to around 18 bases of DNA. And therefore, while designing primers, it is ensured that the primer is not less than 18 bases in length in any case. At the length of 18 bases, the random chance of finding a second perfect complementary site on the genomic DNA, let's say human genomic DNA, 3.2 billion base pairs, is minimal. So a 18-base primer that has not been designed to a repeat sequence in the human genome would normally be absolutely specific and would not have a second perfect priming site on the genomic DNA. And then, of course, you know the base complementarity rules: adenine pairs with thymines forming the double hydrogen bonds, and guanine pairs with cytosine, or vice versa, forming triple hydrogen bonds. So we'll now look at what complications set the upper limit of the length of the primers to be used in a PCR reaction. There is also an upper limit to primer length, and this is roughly around 27 bases. So what are the conditions that set up this limit on the upper length of the primer is what we're going to see next. As the primer length increases, there are two types of complications that become more frequent. First, of course, is the formation of hairpin loop, where a portion of the primer bends over itself and forms hydrogen bonds within its own sequence, forming what is known as a hairpin loop. Important complication that can happen is what is known as the formation of primer dimers. So primer dimer, as the name indicates, there are two molecules of primers that randomly self-anneal with each other because there are regions of complementarity between them. And now what is going to happen is, one, this primer set is not available for PCR. Second, a part of PCR is happening here. This 3' end will be extended based on what are the nucleotides here. Complementary nucleotides will be added here. Likewise, this 3' end will be extended based on what are the complementary nucleotides that are present here. Since the two primers have annealed to each other, they are not now available for a PCR reaction. So this decreases the effective concentration of primer that is present for actually annealing to the targeted site on the template DNA. So therefore, very long primer lengths are avoided. So there is an upper limit to the primer length, which is around 27 bases. The longer the primer, greater the chance for secondary structure formation. Hairpin, primer self-dimer, primer heterodimer can form, and that can impact the efficiency of your amplification and not give you the exponential amplification that you would have expected. And if the primers anneal to themselves or anneal to each other rather than anneal to the template, the PCR efficiency decreases dramatically. Another important criteria for specific annealing is what is known as melting temperature, or TM, of the primer. TM represents the temperature at which 50% of the DNA strands are single-stranded, that is, they're not hybridized to the primer, and the remaining 50% have hybridized to the primer. TM is characteristic of the primer-based composition. And if two primers are of same length, but one has a higher GC content as compared to the other, the one with higher GC content would have a higher melting temperature. And one of the very common formulas which gives you an approximate idea of what is the likely melting temperature of a primer is given by this formula, where Tm, or melting temperature, is equal to 4 into total number of G plus C plus 2 into total number of A plus T. Right? So this is the formula that we commonly use. It gives you an approximate estimate of the melting temperature. And here, as an activity, I ask you a question to calculate the melting and annealing temperature of the following primer. So this is the sequence here. Basically, you have to apply this formula of Tm equals to 4 into G plus C plus 2 into A plus T to get the actual annealing approximate idea of the annealing temperature of this primer. So many a time the annealing temperature and melting temperature are used as synonyms, but they are not actually synonyms. The annealing temperature is slightly lower as compared to the melting temperature. Annealing temperature, or T-anneal, is the temperature at which the primer hybridizes to the template DNA. And this can be derived from Tm very easily, because the calculation is very simple. Generally, T annealing, or temperature at which the primer anneals to the template, would be around 4 degrees or 5 degrees lesser than the melting temperature of the primer, right? And therefore, you already know how to calculate the melting temperature. This is 4 GC plus 280, and subtract 4 from that, and you would get an approximation of what is the annealing temperature for the given primer with a given composition, right? To avoid non-specific primer annealing, Tm of 55 to 65 degrees centigrade is preferred. The higher, the better, right? So remember, when you do a PCR, you have primer in pairs. You have a forward primer and you have a reverse primer, and the two primers must be compatible to each other. By compatibility, what is meant is that the melting temperature of the two primers should not be more than 4 degrees centigrade away from each other, right? Because in the PCR tube that you set the reaction, there is going to be only one single temperature, which would be somewhere in between of the two primer pairs. The maximum difference allowed is around 3 to 4 degrees centigrade. And of course, the closer the Tm of the two primers to each other, the better it is. So let's now summarize the thumb rules in primer designing. Number 1, the primer length and composition has effects on hybridization specificity and melting and annealing temperature of the primer. Second, the longer the primer, the more specific it is, but there are limits to what length a primer can have before other complications come in. So therefore, for perfect annealing, you must have a long primer, but that long primer must be within a certain range of length, which is around 18 to 27 bases. Right? Third, the longer the primer, the higher the melting annealing temperature. And we have already discussed that the primer must have a minimum length of 18 bases and no more than 27 bases in length, so as to ensure that it is specific, and also to ensure that no secondary complication in terms of formation of hairpin loop or in terms of formation of primer dimers occur during the PCR reaction. So what is also important is that overall a random composition of the primer is preferred, random based composition of the primer is preferred. There should not be regions that are rich in AT or regions that are rich in GC. There should be an even distribution of different types of nucleotides on the primer. The GC content of the primers must be between 40 to 60%. This will give the right annealing temperature for ordinary PCR reactions and will give appropriate hybridization stability. Last but not the least, people prefer to have what is known as a GC clamp towards the 3' end. So the GC clamp essentially means that the primer must end at the 3' end with a G or a C, so as to have a strong hybridization with the complementary nucleotide on the template DNA. And I have already shown you if there is no complementarity at the 3' end, then the primer is not extended at all. So the 3' end of the primer should preferentially end in a G or a C. This ensures efficient hybridization and extension, since G or C form more stable triple hydrogen bond as compared to A or T. So now, since I've been talking about the primer for such a long time, let me ask you a basic simple question in PCR again. So which end of the primer is extended by DNA polymerase, the 5' end or the 3' end? So next, it is said that primer designing is an art, and initially during the days of PCR, when the PCR just started, people used to do it manually. Now, fortunately, of course, there are enough softwares and computer programs that would allow you to design primers and also tell you the likely qualities of that particular primer. So let's say here is a template DNA. This is a larger DNA of which we are interested in this region. This is our gene of interest. So ideally, what we would want to have is that the 5' end of the forward primer is at the start of the gene, and likewise the 5' end of the reverse primer is at the end of the gene. And these should then give us the amplification that is desired. However, you remember that we have defined certain rules for primer designing. The length of the primer must be anywhere between 18 to 27 bases. So the primer properties that you're looking for, number one, primer has to be unique. It must have, the template must have only one. The template must have one and only one position where the primer has absolute complementarity with the template DNA. So the perfect primer complementary sequence must appear only once in the template DNA. And to ensure this, you have a primer of length at least 18 bases, and the maximum length is around 27 bases, because beyond 27 bases, the primer can form secondary structures like hairpin loop or primer dimers. Then, of course, remember, for any given amplification, you need two primer pairs, so they have to be compatible to each other in terms of the temperature that is required to perform the PCR. So the compatibility between the primer pairs is of absolute importance. Then, of course, the primer composition, anywhere between 50 to 60% of GC content would allow for stable hybridization. So therefore, the GC content must be 50 to 60%. However, a 40% GC content would also work normally. Right? Then, what also must be ensured is that the primer are not designed to a repeat sequence. Let's say, for example, you talk of the human genome. So human genome has a particular repeat element called ALU element. So ALU has around 1.5 million copies in the genome. So by mistake, if you design a primer to an ALU element, then your primer is going to 1.5 million different sites within the same template to give you an amplification, which will be then non-specific. So therefore, primers must not be designed to repeat sequence, and to avoid that, you can first annotate your sequence using repeat marker, and then take regions which are not repeat for designing our primers. And we've already talked of melting temperature. The melting temperature for primer should be anywhere between 55 to 65 degrees centigrade. A temperature lower than 55 may allow for non-specific annealing, and a temperature higher than 65 may not allow any annealing at all. And then, of course, you have to avoid formation of secondary loops or primer dimers as far as possible. So given that we want a primer to satisfy all these qualities that I've listed here, sometimes it may not be possible to design primers exactly at the start and end of the gene, and instead you may end up designing the primer somewhere in the flanks. For example, let's say here, right? So now you have your gene of interest, which will be amplified, of course, but then there is a bit of an extra sequence also that gets amplified along with the gene of interest. So as I said, the art of finding the most optimum primer pairs to amplify the target is known as primer designing, and is now done using primer designing softwares, right? And I've already told you the most common software that we use is Primer 3. It is also available as a web-based graphic user interface, and also it can be downloaded and installed for a standalone version which can work on command line. So what we're going to now do is to look at why exponential amplification cannot happen if you have a single-stranded DNA and one primer to do your polymerase chain reaction. So here you are. This is your single-stranded DNA that you have, and you have a single primer which is complementary to the end of this template DNA. And you start with first round of your PCR, and the arrow again represents the 3' end, and the blunt end represents the 5' end here. And now when you start with the PCR, so the primer hybridizes to the template DNA strand, which is single-stranded here, and then the DNA polymerase extends it, so you have your extension. Finally, you have a double-stranded DNA here, which is then denatured in the next cycle. So we are now into cycle 2, this gets denatured. And after denaturation, again, the primer can hybridize only to the blue strand here. So it goes and hybridizes to the blue strand, and then gives you an extension, giving you the second complementary strand. And then again, if you move on to the third cycle, so you denature it. And after denaturation, again, you have your 3 DNA strands, 2 of which are complementary. One was the original DNA strand, and again, the primer hybridizes to the original DNA strand because it is only complementary to the blue strand and gives you amplification again. So if you look at the analysis of these extensions that you get, after the first cycle, you have 1 red strand. After the second cycle, you have 2 red strands. And after the third cycle, you have 3 red strands. And there is only 1 double-stranded DNA after the completion of the cycle. So therefore, this is not what is exponential amplification. And therefore, in cases where you have a substrate that is single-stranded, for example, the mRNA, it is first converted into cDNA, and then the cDNA, which is double-stranded, is then taken forward for amplification, right? So here you are, exponential amplification does not take place in PCR with single-stranded nucleic acid molecule and 1 complementary primer, and I've shown you how this works out here in this example here. For downstream experiments, the RNA is first converted to cDNA for 2 reasons. Number 1, RNA is extremely unstable, and therefore, if you want to perform any experiment on RNA, it has to be first converted to cDNA, which is a lot more stable. And one of the reasons why the genetic material switched from RNA, which was largely believed to be the original genetic material, to DNA was the stability of the DNA. And then, of course, the second important point is that exponential amplification can only happen when you have a double-stranded DNA and 2 primers to start your PCR reaction. So therefore, when you work with RNA, first the RNA has to be converted to cDNA, and after it is converted to cDNA, it is then taken forward for quantification using exponential amplification. So this is what we're going to see next. There are 2 types of polymerase chain reactions that we're going to talk of in the next slide. One is what is known as reverse transcriptase PCR, where we are going to convert the mRNA sequence into a complementary DNA sequence, and this complementary DNA sequence can then be amplified. And while amplifying, you want to see real time what is the level of amplification, which is basically done by what is known as real-time PCR. So we're going to see how this works out in the next coming slides. So here is an mRNA molecule, and this could also actually represent the entire transcriptome or all expressed messenger RNAs in a cell. And you know that most of the messenger RNAs in a cell end up in a poly A tail, and that poly A tail can be used as a handle to amplify and convert this mRNA into a double-stranded DNA. The process is known as formation of complementary DNA. And basically what we use here is an oligo-DT primer. And then with the help of reverse transcriptase, you are able to add complementary DNTPs depending on the nucleotides present on the mRNA sequence here. So this is how you synthesize the first DNA strand. So what you now have is an RNA DNA hybrid, and in the next step, you basically remove the RNA strand, and in place of that RNA strand, you synthesize a DNA strand, which now makes it completely double-stranded DNA. We'll not go into the details of how this is done, except for the fact that usually you use RNase H that can partially dissolve the RNA strand, and the steps that are left allow you to extend and add complementary nucleotides here with the help of DNA polymerase. What you can also do is you can add an adapter at the 3' end of the first strand, and then use a complementary primer to that adapter to synthesize the second strand of DNA. The mRNA strand is replaced by a complementary DNA strand here, right? So this is now finally your cDNA, which is basically a double-stranded DNA with the same sequence as was present in the RNA. Now this DNA, this molecule can be used for amplification. So I'm sure you remember from the previous slides that if you start with X molecules of DNA and perform PCR for N cycles, the total DNA molecules on completion of PCR is X into 2 raised power N. So here in this case, again, depending on what is the original number of mRNA molecules for a specific gene, you would get as many number of complementary DNAs to begin with. So you now use primers to amplify the cDNA. And what you would also be interested in would be to see in real time what is the extent of amplification that happens for your cDNA molecule. So to be able to quantify the extent of amplification as it happens is the essence of real-time PCR. So what we do essentially is we use fluorescence to have a real-time check on what is the extent of amplification that is happening. Most commonly, the dye that we use is cyber green. Cyber green can basically bind to both single-stranded DNA and double-stranded DNA, but preferentially it binds to double-stranded DNA. So cyber green has an absorption maxima or can be excited with blue light, and then once it is excited, it can emit as an emission maxima of 520 nanometers, which is basically the green light. So the extent of fluorescence that comes upon excitation of the cyber green after every cycle can give you an idea of what is the extent of amplification that is happening. And also, if you have two different samples, you can compare the extent of fluorescence coming from each other. So here is your initial molecule of cDNA. Here is your starting with the PCR. You have one cDNA molecule, and the cyber green is bound to it, but it may not be, but the system may not detect it. Then as you move on, after the completion of every cycle, the DNA doubles up, and therefore the amount of cyber green that bounds to these DNA molecules also increases and comes into the range of detection. And then, of course, as you move along, you have further increase in the amount of double-stranded DNA. After completion of every cycle, the amount of DNA is going to double up, and therefore the amount of cyber green that binds also doubles up, and that allows you to detect the increase in the number of DNA every cycle as we move into the exponential phase of PCR. So the intensity of fluorescence increases with every cycle and can be monitored in real time, allows for quantitation and comparative quantitation of DNA. So if you have two different samples with different concentrations of DNA, their comparisons can be done. One can also find out what was the initial difference in terms of the amount of DNA present in the two samples by using the real-time PCR. So what are the essential components of real-time PCR? So most of the components are exactly the same as what we had begun with when we were doing the normal PCR. So you'll have DNA or cDNA, you'll have primers, you have polymerase, you have magnesium ions, you have DNTPs. But the most important thing along with all of this, the difference with the conventional PCR is that you also have a fluorescence reporting system. In the initial days of real-time PCR, we use cyber green. Now, of course, we have more complex reporting systems like TACMAN probe, beacons, and scorpions. TACMAN probe, beacons, and scorpions will be discussed elsewhere. We stay with cyber green for now. There are certain important parameters that one needs to check when you're doing real-time PCR to give an idea of comparison of two samples. One of these values is what is known as the CT value. The CT value is the number of cycles taken to reach a specific amplification level, and that amplification level is actually reported by the fluorescence of the fluorescent marker that you use. And then the other important value is delta CT. Delta CT is the difference in the number of PCR cycles taken to reach the same level of amplification by two different samples. So if there are two samples that are comparing, the ratio of A by B can be given by 2 raised to power delta CT, right? Where delta CT is the difference in the number of cycles taken to reach the same amplification level, and 2 raised to power delta CT will give you the actual ratio of the initial concentrations of A and B. So now let's follow it better with an example. So here is a simple example of quantitative PCR. You have two samples, A and B. The initial number of molecules for A is 4. The initial number of molecules to begin with is 1. For B, the final number of molecules that we're looking at is 64 for both A and B. The number of cycles required by A to reach 64 level is what is known as CTA. Likewise, the number of cycles required for B to reach 64 molecules is what is known as CTB. And how do we monitor the levels of amplification? We monitor the levels of amplification by measuring the fluorescence that comes from on the completion of the PCR cycle. So let's now calculate for A first, and then we calculate for B. And since we're starting the experiment, we know that the original ratio of A to B is 4:1, because A has 4 double-stranded DNA molecules, while B has 1 double-stranded DNA molecule at cycle 0 of the PCR. So this is 4/1 or equivalent to 4, right? So now we calculate the CTA value for sample A. So here is solving for A. So 4 into 2 raised to power CTA equals to 64, which means 2 raised to power CTA equals to 16, or CTA, or the CT value for A equals to 4 here, right? Next, we solve for B. So again, you know, the final number of DNA molecules at cycle N is given by the formula x into 2 raised to power n. So here x equals to 1. So 1 into 2 raised to power CTB equals to 64, which means CTB equals to 6. So now the CTA is 4, CTB is 6. So now we calculate the delta CT. What is delta CT? The delta CT is the difference in number of PCR cycles taken to reach the same amplification level by two different samples. So delta CT-AB, which means you're comparing A and B together and you're calculating delta CT here, equals to 6 minus 4 equals to 2 here. So now if you put it to the formula of finding the ratios of the initial samples, initial concentration of molecules in the two samples. So here you are, ratio of A to B equals to 2 raised to power delta CT. We know that the delta CT value is 2. So therefore, putting delta CT value here, it gives you the ratio of A by B equals to 4, which is the same as what we know because we started the same number of molecules here. So this is what is known as quantitative PCR. Quantitative PCR allows you to quantitate the amount of DNA that is getting formed every cycle, and using CT and delta CT, you can go back and calculate the initial ratio of two samples to begin with. So this is your quantitative PCR, and I'm sure you already know that the primary test for checking for SARS-CoV infection is the real-time PCR. So this is how you do your real-time PCR and quantitate the amount of DNA that is present in your test sample with respect to a control sample. So with that, we come to the end of the lecture on PCR. There is a lot more to PCR than this, of course, and as and when we have time, we'll discuss more about PCR, particularly the tinkering that you can do with polymerase chain reaction that allows for a lot more innovative methods of biological assays. So we'll discuss those as we move along during the course.